actually talking on behalf of something called the Insignia Consortium. And the Insignia Consortium actually consists of 16 partner institutions from 12 European countries. And here we all are, and we're actually quite a varied bunch of people. We are bee research scientists, we are beekeeping extension specialists, we are molecular biologists, we are analytical chemists, we are modelers, we are statisticians, and we've also got some social scientists on board to try and understand the motivation behind why beekeepers would want to take part in a citizen science project. But before I get on to Insignia, I want to talk very briefly about a previous citizen science project that a lot of the consortium were involved in. This was called the CSI Pollen Project, standing for Citizen Scientists Investigation on Pollen Diversity Forage Available to Honeybees. And this was done through the auspices of the Colos Association that is so active in trying to understand all the reasons why honeybee colony losses may be occurring and why colonies might be dying. And one of the possibilities there is that there are areas where honeybees are foraging on a very poor diet in places with a lack of biological diversity in terms of the plants that they can forage on. And so it seemed logical that there's a possibility that maybe high colony losses occur in those places of low diversity, and that places with a high diversity have lower losses. And one way to study that on a large scale is to get beekeepers to do the work for you. And so what we did was we recruited beekeepers, and they needed to have 3 honeybee colonies available, and every 3 weeks they put pollen traps on their colonies and collected a pollen sample. And the key thing here is that all these beekeepers were collecting their samples at exactly the same time in the countries and places involved in the project. And then they were taking a 20-gram subsample of the pollen they collected in this high-tech piece of equipment here, which happens to be a standard honey jar lid, which holds roughly 20 grams of pollen. And then all they were doing was laying it out on a white tray, and they were dividing the pollen using a little brush or something into the number of different colours available. And this is a proxy for the diversity of plants that are available. And we know, of course, that it's not an exact science in that you find certain plants will produce pollen of different colours in different circumstances, and equally the same colour may actually be several different plant species. But nonetheless, if everyone does the same thing, it's a very good guide to pollen diversity. And the last key thing about what we were doing was we had a web-based system using Lime Survey, which meant that the beekeepers were putting their data onto their own computers using a unique code that was sent to them, and that went straight into a central database. So over the 3 years of the study, it proved to be a very successful citizen science project from terms of the participants, and it was actually carried out in 27 European countries. A total number of 586 citizen scientists took part, of whom 316 were regular enough to actually produce really useful data. I personally had 71 beekeepers collecting samples in England and Wales. And over the course of the study, 17,464 samples were collected. And this shows the possibilities of a citizen science project, because there is absolutely no way that an individual research institution would be able to do work on this kind of scale. And this is where they all were, and you can see that they're very spread out in many European countries. It produced lots of results, and I'm not particularly going to talk about them today. Here is Robert Brodschneider talking about the research at the EU conference a couple of years ago, and a refereed paper with the results will hopefully be available fairly soon, and you can find out about it. I'm just going to show you one graph here, which shows that overall the answer was 4. In other words, we found that the bees consistently have about 4 major pollen sources that they collect from. And an interesting thing about the project was that this was remarkably constant over the 2 years, and over the different countries, and over different land uses, which was not entirely what we were expecting. And this suggests that even in places where there was a very low diversity of forage available, bees are going to quite a lot of trouble to find a range of pollen sources for themselves. And this, of course, throws up all sorts of other interesting questions. So having got quite a long way with this project, we had a meeting in Glasgow in February 2016, and we were discussing how the project was going and where we might go from there. And we thought, well, we've got all these thousands of pollen samples sitting in freezers all over Europe. What could we do with those? And then we thought, well, of course, they could be tested for the presence of pesticides. And for about 5 minutes, we were really enthusiastic about this idea. But then we realised that there were some pretty fundamental difficulties in that we had no control over how these samples had been collected. There were serious possibilities of contamination between hives, between samples, and we didn't really know how they'd been stored. And the real fundamental problem is that pollen, being a biological material, is incredibly prone to decay and degradation by bacteria and fungi if it's not kept under very closely controlled conditions. So we gave up that idea. But then, last year, we spotted that the European Union had put out a call for a research project doing exactly that. And they wanted people to put in a proposal for a citizen science project to get beekeepers to collect pollen samples for pesticide analysis. So given our experience, we thought, well, let's apply, and we are pleased to be accepted. And I just want to think for a minute about why honeybees are good as environmental monitors, and this follows on very nicely from the previous talk. This is a Google map of this white dot here is our lab at the University of Sussex in southern England, and we're located on this range of very low hills which are called the South Downs, which are now a national park and hopefully being restored to a more natural state, and there's a very important national nature reserve close by. But we've also got the urban area. This is it's actually a city or large town of Brighton and Hove here. We've got a small town of Lewes here, and then north of the South Downs we've got agricultural land that's arable land. So our bees, foraging over the kind of distances they do, have quite a range of different things available to them. And I'm just going to very quickly show you results from a project that a former colleague of mine, Maggie Couvillon, did when she was based at the lab. And she and her colleagues spent quite a number of years decoding waggle dances and then plotting the results on the map. And it shows the difference that you get through the year. So in March, the bees are foraging on the university campus and very close aside, and April, May, June, July, August, you can see they're going a long way away because the colonies are large at that time and because there's not very much forage around. They're going into the town, they're going around, and then towards the end of the season you can see they're foraging on different things. So this opens up the possibilities that those bees will be picking up all sorts of environmental contamination from a variety of sources which make them ideal. So the project that we came up with is called Insignia, and The acronym stands for Citizen Science Investigation for Pesticides in Agricultural Products. And this is our, yes, it's a very tenuous collection, I have to say. And this logo simply shows the different techniques that are involved in it. And we had a meeting in the Netherlands last November, and then we're actually actively in year one. So really, the aims of the project are to develop a citizen science protocol. So we're not actually aiming to do a research project as such. We are developing a protocol to use honeybee hive products to explore pesticide contamination and relate this to the crops on which bees are foraging. And this will be used to model the risk to bees of different habitat types. So in year one, we're sampling in just 4 countries: Austria, Denmark, Greece, in the UK, and in each country we picked 4 sites, and we aimed for 2 low risk, that is in hopefully semi-natural vegetation, organic farmland, something like that, or 2 high risk, which would be arable farming in each country, and 3 colonies on each site, and there they are. So, at each of these sites, as we were asked to do by EU, we have pollen traps, much as in the CSI pollen project. But we're still mindful that those objections we thought of a few years ago are perfectly valid, and we are concerned that those pollen samples decay very rapidly. So we're using 2 passive systems as well. So some of the colonies have something called a bee-held tube, which was developed by colleagues in the Netherlands, and this is simply plastic tubes here that go at a modified entrance on the hive, and bees come out of these tubes and they have to go in through these plastic tubes that are lined with polyethylene glycol, which will pick up chemicals and pollen grains from the bees' body as they pass through. And then the second passive device is something called the Apistrip, developed by colleagues in Spain, and this is just a plastic strip, a bit like an Apistan or Beverol strip, and this is coated with a compound called Tenax, which is particularly good at picking up and absorbing chemical contamination. And then the final thing that we're doing is we're collecting samples of bee bread from within the colony using this little natty little device developed by some of our colleagues in Italy, although actually we're often using pieces of drinking straw which work quite well. So, once a fortnight, these samples are being collected, and the samples are split for analysis in Portugal for the botanical origin of the pollen using DNA barcoding, and then the remainder of the sample is sent to 2 labs either in Spain or Greece for contamination by authorised and unauthorised substances. So we're looking for all agricultural chemicals that are authorised in these countries, and unauthorised ones that people are known to be using, and this also includes the kind of things that beekeepers are putting into the hives themselves, like varroa treatments. In year one, as I say, it's 4 countries. Year 2, we will, having hopefully refined the protocol, we will be adding Ireland, France, Italy, Belgium and Latvia to the list. I'm going to finish by some very, very preliminary results which only came in a couple of days ago. These are just from Denmark, and these are just from the Apistrips, which, as I say, are these little plastic strips here. This is just an example of the kind of data. So this is 4 apiaries here, the different sampling dates here, and this is just the number of compounds that have been picked up in these samples, and it depends, what you're setting as a threshold concentration. This is taking out the lower ones, and it shows that you do indeed get compounds on these strips. And this site over here was one of the high-risk agricultural ones, and this is an organic site over here on the right. So things are in the right area. And again, some more graphs. These are specific chemicals. All these ones up the top here are fungicides, and you've got 2 neonicotinoids turning down here. This is imidacloprid. This, in fact, is Fleming's apiary here, and it's interesting to speculate exactly how some of this contamination may have got there. So, very immediate conclusions. We do get differences between the land use. The organic sites seem to have lower incidence than the others. These chemicals really could have come from anywhere. And the key thing for us, our responsibility, I think, is that we need to be very cautious about how we interpret these results, particularly when telling the beekeeper that, okay, over the course of the season you had 15 different compounds in your colonies. What does that mean? The so what question is very important in these sorts of studies. So really watch this space for future thing. Where can you watch this space? Well, here is our website, which has a blog on it with what's going on. This is our Facebook page, and we've already published articles about the project in a number of different languages. And I must thank the European Union for funding this study, and here is where you can find information about the project. Thank you. Do we have one very quick question? No?
How many different compounds were you testing for?
It's a very long list. I don't have the complete list, but you know, it's many compounds that are being tested, and hopefully it includes, you know, all the chemicals we can think of. It's a very thorough set of compounds.